Question time. If someone asks that why does membrane fall apart when it's too hot, and why does the cell membrane freeze when it's too cold? So the membrane, they do fall apart when it's too hot, and they do have a freeziness when it's too cold. The reason being same is because they have a unique property which is known as membrane fluidity. So there are many different factors that affect the membrane fluidity. For example, temperature, cholesterol, saturated, unsaturated fatty acids, which we are going to discuss next. So if someone asks you that why do the membrane, they also have the effect of hotness or coldness, that's because of their fluidity nature, membrane fluidity nature. So let's discuss about the factors which affect those membrane fluidity. So you can take a movement and write down the factors affecting the same: temperature, length of the fatty acids, degree of saturation of fatty acids, and amount of cholesterol which are present. So how the temperature affect the membrane fluidity? At low temperature, there is a less kinetic energy of the phospholipid. Therefore, there will be less movement or less diffusion, the lateral, the rotational or the flip-flop movement. Therefore, they will have the less membrane fluidity when the temperature is low. On the other hand, when temperature is high, of course, they have the high kinetic energy. The high movement or diffusion will be there. Therefore, the membrane fluidity increases. So, temperature is directly proportional to the membrane fluidity. Next up, we have length of the fatty acids. How do they affect? So first one is if we have short chain of fatty acids, which is this one. So if we have short chain of fatty acids, they have less hydrophobic and van der Waals interactions, which is shown right here through this red arrow. So the membranes become less rigidity and there will be more fluidity. So if they have the less interaction with among itself only, then of course they won't be that rigid, they won't be that close. Therefore, there will be more fluidity. They can move along freely. If short chain of fatty acids are there, remember, number of hydrophobic and van der Waals interactions are less. On the other hand, if you have long chain of fatty acids, of course there will be more hydrophobic and van der Waals interactions. The bonds in number will also be more, therefore they will be more rigid because they are now more close if you see to this picture itself, so they are less fluid. So longer the fatty acids chain, the less the fluidity. So length of fatty acids is inversely proportional to the fluidity. Next up, we have the types of, first of all, that is the tails of fatty acids. So we have two types of fatty acids tail: saturated and unsaturated. Saturated are the ones which have no double bonds in them, and unsaturated are the ones which have double bonds in them. So that's what the types of the fatty acids tails are. Now, depending on that, also membrane fluidity is affected. So if we see first of all with the saturated fatty acids, as there are no double bonds, they are straight fatty acids chain. So that looks like this, the saturated one, they are straight, no double bond present. Unsaturated fatty acids, they do have the double bond, and because of that they are curved or kinks are present. Example of unsaturated fatty acids are linolenic acid, and myastic acid is the example of saturated fatty acids. So we'll see how do that affect the fluidity. So here, the third one. If we have the saturated fatty acids, they are straight, so there will be tight association with the neighboring fatty acids. So their membrane fluidity will be weak. They do have the tight association in between them. But on the other hand, the unsaturated fatty acids, since they are banded, they have kinks present in them. They are loosely associated with the neighboring fatty acids. Thus, they do have the membrane fluidity. So, higher the number of double bond present, higher the fluidity. That is, the more the unsaturated fatty acids, the more is the fluid, because they will lose that interaction. So if you see from this diagram, compare the diagram for the C. That's why I kept the diagram so that you can check out how rigid and how fluid they are. Next up, we have the role of cholesterol in membrane fluidity. So cholesterol is relatively small molecule, but it is just like the phospholipid. Okay, a cholesterol molecule do have the hydrophilic head right here, and they do have the hydrophobic tail. So they fit nearly between the phospholipid molecule with their head at the membrane surface like this. So this is the structure of the cholesterol. If you see the OH group, hydroxyl group make the head polar and hydrophilic, and they are attracted to the phosphate head on the periphery of the membrane. And the carbon rings, it's not classified as fat or oil, but cholesterol is a steroid because of their particular structure. And here they have the non-polar hydrophobic tail, which is attracted to the hydrophobic tail of phospholipid in the center. So that's how the cholesterol will just fit nearly between the phospholipids molecules. And let me tell you, the cell surface membrane in animal cell contain almost as much as cholesterol as phospholipids are present. But cholesterol is much less common in plant cell membranes, and they are absolutely absent in prokaryotes. Now, what is the main function of the cholesterol? So let me first tell you that cholesterol, it is known as a buffer. It is a kind of a buffer. Okay, it allows a fairly stable and normal level of fluidity. Basically, it stabilizes the fluidity. Now, how do they do that? Let's discuss. So, if you see the cholesterol, when it is too hot, what happens is, when they are too hot at higher temperatures, they just stabilize the membrane. That is, the steroid cholesterol has a different effect on membrane fluidity at different temperatures. At warm temperatures, such as 37 degrees Celsius, cholesterol that restrain the movement of phospholipids. Because as we have discussed, that as the temperature was more, the fluidity was more. Why? Because kinetic energy will be more. But then what happens if the temperature keeps on increasing? They will just move apart. They will become too fluid in nature. If you see this picture, if still the hotness or the temperature is increased, this phospholipid bilayers or the components which are present, they just move apart to become too fluid. So cholesterol, they just restrain that movement. They hold them together. Okay, they won't allow them to move too far from each other, otherwise it will become too much of fluid. Now, when there is lower temperature, at that time what happens? That is the cooler temperature. It maintains fluidity by preventing it from tight packing. As you see, when it is too cold, the phospholipids, they just freeze together. They come together, and the rigidity will increase. But thanks to the cholesterol which are present in between them, they just prevent this tight packaging of this particular phospholipid bilayer when it is too cold. So this third picture is regarding the same too hot. If there is too hot or too cold, they maintain the ideal fluidity like this since they are present. They won't allow the phospholipid to move too away from each other, and they won't allow them to come too closer to each other. That's how they are known as buffer fluidity. They are the buffer for maintaining the fluidity of the phospholipid bilayer. So that's the main function of the cholesterol. Since they have the similar structure to that of the phospholipid, they do have the hydrophilic head and a hydrophobic tail. They just act similar to that of the phospholipid. That's the main role of the cholesterol. So if you see right here in this picture, this is too fluid, this is too viscous and rigid. So what happens is cholesterol within the animal cell membrane right here in this picture. Cholesterol that reduces membrane fluidity at moderate temperature by reducing phospholipid movement, but at lower temperatures it hinders solidification. Basically, they won't allow them to come too closer to each other to prevent tight packing. That is solidification by disrupting the regular packing of the phospholipid since they are present right here. So there could be some related questions for the same. What enhances membrane fluidity in the membrane with unsaturated fatty acids? So what basically enhances the membrane fluidity when there are unsaturated fatty acids? Because they are spread apart, so there will be more room for the movement of the phospholipid. So when they are unsaturated, that enhances the fluidity. Second is what causes membrane with saturated fatty acids to be more viscous, or they will be more resistant to the flow since they are packed together. When they are viscous, they are packed together. That is what causes the saturated fatty acids to be more viscous, which we have already discussed. Now, cholesterol. They are known as fluidity buffer. So why does cholesterol, there will be two questions related for the same. First one is why does cholesterol reduces fluidity at moderate temperature? That is when the temperature is warm. It gets in the way of the phospholipid which are moving around. When they are moving apart, they just gets in their way, they hold them together. You can just imagine like this, two friends, if they are moving apart from each other, and the third friend, which is just like them, they also have two hands and two legs, they hold them together. Please don't move apart. Like that only cholesterol do that. They won't allow these two friends to move apart from each other. And second one, why does cholesterol hinder solidification at lower temperature? That is when the temperature is too cold, why do they prevent the tight packaging of this particular phospholipid bilayer? Answer for the same is it gets in the way and prevent phospholipid from the packaging. Again, in both the situation, they get in the way because they are present in between the phospholipid bilayer because they have the same structure as that of the phospholipid. So this could be the related questions for with respect to the fluidity of the membrane. You can just pause a video right here and take a note of the same. Apart from that, the other functions of the cholesterol. It is important for mechanical stability of the membrane, as without it, membranes will quickly break and cell will burst open. Hydrophobic region of cholesterol molecule help to prevent ions or polar molecule from passing through the membrane. Next is, it is important in myelin sheath, the myelin sheath which are present in the neurons around the nerve cells or neurons, where leakage of the ions would slow down the neural impulse. So there, the cholesterol part is very important. So now as you see from this picture, the presence of the cholesterol, how do they affect the fluidity? The one which we have already seen. So you can just take a note, less amount of cholesterol, this green one is the cholesterol. less hydrophobic interaction with the fatty acids, so membrane becomes more fluid. If less cholesterol is there, there will be more fluidity because there won't be cholesterol present. When high cholesterols are present, there will be more hydrophobic interactions. The membrane become less fluid. When number of cholesterols are present more, the phospholipid bilayers, they won't have the chance to communicate with each other and there will be less fluidity. So factors affecting fluidity, if someone asks, with respect to presence of cholesterol, low cholesterol content, more fluidity, high cholesterol content, less fluidity. And apart from that, they maintain the membrane fluidity. If someone asks the questions, we have already discussed that, but they act as a buffer. So that's it with the membrane fluidity, factors affecting them, the roles of the cholesterols, and what do you mean by the fluidity. So I hope this video was helpful to you, and if that's so, please subscribe my channel and click the like button, and you can also comment in the comment box for any of the queries, or if you want some more videos, if you're having any difficulties, so you can just give me any suggestions in the comment box. Thank you so much, and we'll see you